Okay, in this video, we are going to look at the excitation-contraction coupling process for smooth muscle. Smooth muscle is a little bit more complicated than skeletal muscle only because there's a lot more ways to get calcium levels to go up in the cytosol. What's the same as with skeletal muscle, and as we'll see later with cardiac muscle, is that increased levels of cytosolic calcium are needed for contraction. Lower levels of calcium mean less contraction. So let's start with just orienting ourselves to this diagram. So this yellow line here is representing the plasma membrane or sarcolemma of the smooth muscle. And on that membrane is that's a sarcolemma, it's not a very clear M, is a calcium ATPase, which is going to pump calcium out. We'll come back to that later. And of course, we have a calcium channel with various gating mechanisms. So as you've read in your notes, it could be a change in voltage, mechanical stretch, or a ligand binding. Or this could be a receptor that's connected to other proteins, and a second messenger system results, or activation of that second messenger system rather, results in increased levels of cytosolic calcium. And then we have our sarcoplasmic reticulum here, the SR, with storing lots of calcium in it, and again, a calcium ATPase to put calcium back. Okay, let's start with something coming along and opening this calcium channel, and let's pretend it's a mechanically gated calcium channel, so some stretch. So maybe we're in a blood vessel or the bladder, and it opened and calcium entered. And when calcium enters the cytosol of smooth muscle, it binds with a regulatory protein called calmodulin. So when there's no calcium in the cytosol, calmodulin just hangs out and doesn't do anything. But then calcium binds to calmodulin. And this is very similar in structure to troponin. And this calcium calmodulin complex activates another protein. So we'll show a arrow here. So there's another protein called myosin light chain kinase. So we have inactive myosin light chain kinase, MLCK, that just hangs out and does nothing until calcium calmodulin comes along, and then we have active myosin light chain kinase. Well, what this does, it's a kinase, right? So it adds a phosphate to something, and what it does is it adds a phosphate to myosin. So here we have myosin light chain kinase. Once it's been activated by calcium calmodulin, takes an inactive myosin and adds a phosphate to it. That's PO4 phosphate. Now we have active myosin. So this is really kind of different from skeletal muscle, where myosin is always capable of being active. It's just waiting for the binding site of actin to be revealed. With smooth muscle, the binding site of actin is always available, but myosin is not active until it has another phosphate added to it. And then once myosin is active, it's able to go through the cross-bridge cycle. So let me just put a little arrow going up here. And then we'll say cross-bridge cycle starts, which is pretty much the same as in skeletal muscle when once myosin has become activated by having an extra phosphate added to it. Well, what stops the contraction? So myosin gets activated, undergoes the cross-bridge cycle. Well, there's another enzyme that is always working, opposing the activity of myosin light chain kinase, and that is myosin phosphatase. And what that does is removes, myosin phosphatase removes a phosphate. So there's a war going on between myosin kinase and myosin phosphatase. So myosin phosphatase is always, always, always active, and it's constantly taking the phosphates off myosin to inactivate it. And myosin light chain kinase is always adding the phosphate to it as long as it's active. So what that means is to have active myosin light chain kinase adding phosphate to myosin and allowing it to undergo the cross-bridge cycle, you have to have calcium calmodulin present. To have calcium calmodulin, you have to have calcium. So in order to have contraction, you have to have myosin kinase more active than the myosin phosphatase. Otherwise, you will have relaxation happening. So for contraction, MLCK activity has to be greater than myosin phosphatase. And I'm going to just abbreviate that activity. So it's really a graded response because the amount of myosin kinase activity depends on how much calcium calmodulin is present, and that depends on how much calcium got let into the cytosol. So you have a graded response to calcium. A little bit of calcium gives you a little bit of contraction. A lot of calcium gives you a lot of contraction. So again, we always have to be talking about, well, how do we stop the signal and stop the contraction from happening? Well, there's a couple of things. Obviously, myosin phosphatase activity is always opposing myosin kinase, so trying to cause relaxation to happen. But if calcium stops getting let into the cytosol, if we close these calcium channels, then calcium, in order to get rid of calcium, we have to pump it out against its concentration gradient. So that means we can pump it out into the interstitial fluid across the sarcolemma using a calcium ATPase, or it can get pumped back into the sarcoplasmic reticulum. So what's the role of the sarcoplasmic reticulum? How does calcium get out of this? Well, it depends. You can have, for example, a protein here that's attached to another protein here, and a ligand will bind to it. That's a ligand, and that will activate another protein that will end up letting calcium out. So a second messenger system pathway can result in calcium being let out of the sarcoplasmic reticulum. And we're not going to worry about the details of those mechanisms, but just be aware that it's not always a ligand-gated channel directly increasing the calcium in the cytosol, but it could be, that's a ligand right there, that dot. It could be a ligand binding with a receptor attached to a second messenger system, resulting in increased levels of calcium. The point is, though, that the more calcium that's in the cytosol, the stronger the force of contraction. Less calcium leads to a lower force of contraction. And there's a lot of therapeutic drugs that influence the state of contraction of smooth muscle, and some of them influence channels, some of them might influence these receptor proteins, some of them might influence the state of myosin light chain kinase or even myosin phosphatase. So the process is a little more complicated than skeletal muscle, but we can make it pretty straightforward if we remember that calcium linked to increased levels of myosin kinase activity, lower levels of calcium with lower myosin kinase activity, and of course, to get contraction, myosin kinase activity has to be higher than myosin phosphatase activity. Okay, hope this is clear. Another place to look at this is in your textbook. Figure 1226 has a slightly different version of this drawing if you want to take a look at that.